Now we have to do this unit of carbohydrate metabolism. Now before starting the actual carbohydrate metabolism, again I'll be taking few basics so that when you do these basics, it will be very easy for you to understand the pathways and cycles in carbohydrate metabolism. So let's get started. First we'll be doing basics. Now, when let's say a question is there and the question is not mentioning fed state or fasting state, then by default you have to consider fed state only because we normally are in fed state, right? Now, in fed state, a particular diet we are taking, this diet mainly contains carbohydrates. That is 60 to 70% of our diet is carbohydrates. All this roti, chapati, rice, pasta, pizza, bread, everything is carbohydrates, right? So 60 to 70% main thing is carbohydrates, for which prototype is glucose, right? And 15 to 20% is the most tasty thing in the diet, and that is fats, and rest is proteins. Rest is proteins. So we have to see that what will happen to these carbohydrates when these carbohydrates are taken. Then what will happen to this carbohydrate in the body? Carbohydrate which is taken, only 50% of it is used for energy. When I'm doing this calculation kind of thing, then I'm taking a general scenario which is happening worldwide, that we have a sedentary lifestyle these days, but on top of it we are taking 2 to 3 meals in a day. That is, very less energy is required for us, but we are taking so much of energy in the body because carbohydrates we are mainly taking, and carbohydrates are for energy, right? So in the sedentary lifestyle, only 50% is used for energy, but rest 50% is stored in the body for future purpose. Out of this 50, 10% is stored in the form of glycogen, which we are using in between 2 meals. We use this glycogen to maintain blood glucose levels, but rest 40% of all the carbohydrates we take. See, imagine 40% of the carbohydrates we take is converted to fat in the body, and this fat is endogenous fat. How endogenous? See, in the diet though we have taken carbohydrates, right? But this fat is getting formed in the body from carbohydrates. So this fat gets synthesized in the body, known as endogenous fat. Endogenous fat gets transported in the form of VLDL. And this fat, which is we are taking in the diet, this fat is exogenous fat, gets transported in the form of chylomicrons. So this is the basic concept of diet, and this concept should be used when some person wants to reduce weight. But a layman is not knowing all these concepts. So what a layman will do, when a layman will do a kind of dieting to reduce weight, then what they will do, they think that we are fatty, so we stop the fat in the diet in our dieting. When they stop the fat, they're just stopping the exogenous fat. But when in this dieting, they are taking carbohydrates only because they are taking chapati, boiled rice, fruits, boiled vegetables, juice, like that. So all this is carbohydrates which they are taking. When they are not taking fats, they're feeling hungry and they're taking more carbohydrates in the diet. So this excess of carbohydrate will be converted to fat in the body. This is known as endogenous fat getting transported in the form of VLDL, and this kind of person, this kind of dieting will in the end increase the weight of the person. And this is a frequently asked question also. See the question, a person on fat-free, carbohydrate-rich diet continues to grow obese, which lipoprotein will be increased in this person? Answer is VLDL, because this kind of person is not taking any exogenous fat, so chylomicrons won't be found in this person's blood profile. But this person is taking excess carbohydrates, which is getting converted to fat in the body, which is getting transported in the form of VLDL. So when you check lipid profile of this kind of person, then VLDL will be increased. So because of this concept, people don't know this concept, and when they do dieting, they end up increasing their weight. So that's why they need professional help. That's why many kind of diets are famous on internet. Many dietitians are famous. Now, one diet known as Atkins diet, Atkins, by the name of person who discovered it. Atkins diet. This question was asked in AIMS. What is Atkins diet? Atkins diet is low calorie. See, calorie concept we have to follow in order to reduce weight. low calorie but also low in carbohydrate. This is Atkins diet, low calorie, low carbohydrate diet. When taken, it will help reduce weight. Now, many people have a misconception that they can take juice in dieting. Juice is very light, so they think we can take. But see, juice is, fruit means fructose. Fructose is a carbohydrate. It will be converted to fat in the body. Plus, on top of it, one more point is there that fructose is most lipogenic sugar. Most lipogenic. Out of all the carbohydrates, fructose is most lipogenic. So when you are taking a glass of juice in dieting, that is the full fattening drink which we are taking, right? So we should not take juice in dieting. We can take fruits because fruit has lots of fiber in it, right? There is a term, thermogenic effect of food. See, this was also recently asked question. See, food is giving us calories in the body. When we are taking food, it is giving calories in the body. But food is also using calories of the body. That is, we need energy to digest that food, then absorb that food from the intestine, then transport that food from intestine to cells. Then in the cells, metabolism of that food has to occur. So we require energy for all this work to be done in the body. And that energy required is known as thermogenic effect of food. So I'll read it. Thermogenic effect of food is energy required to digest, absorb, transport and metabolize the food. This thermogenic effect of food is also known as SDA, that is specific dynamic action. And this thermogenic effect of food is maximum for proteins, then for carbs, then for fats. So when we will take proteins in the diet, protein itself will use so much energy of the body that it will help reduce weight. Even if we are in sedentary lifestyle and we want to reduce weight and we don't have time for exercise, then we can do this kind of dieting, taking more proteins and less carbs and less fats in the diet. So this will help reduce weight. That's why these days everybody is shifting to protein diet these days. They're so famous, right? Now in these basics, now I'm also going to take few basic points of enzymes. Otherwise enzymes topic I'm taking after carbohydrates. Right now I'm going to discuss basic points of enzymes because these basics of enzymes will help you understand the pathways and cycles so easily, because each pathway and each cycle is nothing but enzymatic reactions. Let's say glycolysis, there are 10 enzymatic reactions. TCA cycle, there are 8 enzymatic reactions. So when you're very clear about the basics of enzymes, then it is very easy to understand those pathways. So this way I will be teaching. So, enzyme classification we do in a sequence OTHLIL. The sequence we do. Why this sequence is important? From number 1 to 6. Why this sequence is important? Because these 6 numbers are known as enzyme commission numbers or enzyme code numbers or simply EC numbers. These days they can ask you EC number of any enzyme and I'll try making it very easy for you. Let's see. What is this full form of OTHLIL in a sequence when we do? This O is oxidoreductase, the enzymes which are carrying oxidation reduction reactions. T stands for transferase, H stands for hydrolase, and there are 2 L here. Number 4 is also L, number 6 is also L. This number 4L is lyase, LY, lyase. Then number 5 is I for isomerase. Number 6 is L for ligase. This number 6L is ligase, right? Now let's see one basic point, that whenever there is a bond, let's see there's a bond. If we have to break the bond, then we have to add water to break that bond. And if we have to make some bond, then we have to use the energy of ATP to make that bond, right? So to break a bond, we need water. To make a bond, we need ATP, energy of ATP to make the bond, right? Tell me, which is that enzyme which uses water to break? The name is telling you. The name is telling you. Hydrolase will use water to break. And which enzyme uses ATP to make? That is ligase. Ligase will use ATP. Ligation means joining. It will use ATP to make. Now there's one enzyme which can either make a bond or break a bond. Both things it can do, but it neither requires water nor it requires ATP, and that enzyme is lyase category. Lyase can make a bond or break a bond, but it neither requires water nor it requires ATP. And now for rest 3, there's no need to write. It is very clear from the name. Oxidoreductase, the enzyme which carry oxidation reduction reactions. Transferase, which will transfer groups, right, from one group. From one molecule, the group is removed. In another molecule, the group is added. It can be any group. And then isomerase enzymes, they will convert various isomers into each other, right? Now, there is one big difference between transferase and isomerase. Tell me, when isomerase work, then molecular formula will be changed or not? Molecular formula will not be changed. When isomerase work, molecular formula will remain same because one isomer is converted to another isomer and isomers have same molecular formula. But when transferase will work, then molecular formula will be changed. That is, a group is deleted from one molecule, added in another molecule. So either addition or deletion, molecular formula is going to be changed. For some enzymes, we are confused that the answer is transferase or isomerase. At that time, you will be using this point to answer, right? Now tell me, if oxygen is added to some compound, then that process is known as oxidation. If hydrogen added, then if hydrogen is added, then it is known as reduction. If electron is added, then also it is reduction. So in biochemistry, consider hydrogen or electron one and the same thing. So in a question, if hydrogen atom is written or hydrogen written or reducing equivalent is written, then you will say it is electron they are talking. And if hydrogen ion is written, then that means it is equivalent to proton. So for making biochemistry concept simple, just cram this thing that hydrogen atom is equivalent to electron, hydrogen ion is equivalent to proton, right? Now there is one enzyme which is involved in almost every pathway in the body, and that enzyme is very important, dehydrogenase. Let's see what this enzyme is doing. Dehydrogenase. What do you get from the name? What is happening? Dehydrogenase means there is removal of hydrogen. Removal of hydrogen means oxidation. So oxidation occurring, let's say a compound A gets converted to its oxidized form, right? So here hydrogen is getting removed. This hydrogen, when it is getting removed, it is not simply thrown into the medium. Some molecule will come here to take this hydrogen, right? Whenever dehydrogenase work, hydrogen is removed. Somebody will come to take this hydrogen. That somebody can be one out of these three things. Either it can be NAD or FAD or NADP, which will come to take this hydrogen. Respectively, they will take hydrogen and will form NADH, FADH2 and NADPH, right? Now this NADH will go into ETC to give 2.5 ATPs. So earlier it was 3 and 2 ATPs, but now it is 2.5 and 1.5 ATP. That is NADH gives 2.5 ATPs and FADH2 gives 1.5 ATP via ETC. But this third molecule, NADPH, this is not taking part in any ATP formation. This NADPH takes part in reductive biosynthesis. I will explain the meaning later, but little bit you can understand. Reductive biosynthesis means anabolic pathway. So this number 3 point is used in anabolic pathway, but this number 1 and number 2 point is used in catabolic pathway. You know, any enzyme, any pathway which gives ATP in the end, that pathway is catabolic pathway, right? I summarize here that whenever dehydrogenase work, then hydrogen is removed, it is oxidation occurring. Somebody will come to take this hydrogen. In catabolic pathways, NADFAD is involved. In anabolic pathways, NADPH is involved. Now this NADPH is very, very important. Many questions are asked on this NADPH. They can ask that NADPH is produced by which pathway and NADPH is used in which pathway. Both things they can ask. So be careful what they're asking, and I'm going to make it very clear on this page. First, see from where it is produced. It is produced from which pathway? The major source of NADPH is HMP, right? HMP is the major pathway for the production of NADPH, but we have two minor pathways also. One is malic enzyme and other is cytoplasmic isocitrate dehydrogenase. Where is this enzyme, cytoplasmic isocitrate dehydrogenase? You know this enzyme is there in Krebs cycle, but Krebs cycle occurs in mitochondria. So this is not the enzyme of Krebs cycle. This is isoform of this enzyme present in cytoplasm known as cytoplasmic isocitrate dehydrogenase, right? So these are the three pathways which are producing NADPH. And now let's see where is NADPH used. I told you it is used in reductive biosynthesis. Biosynthesis means anabolic pathway. In anabolic pathway, actually, whenever we will do some anabolic pathway, we will always encounter some reductase enzyme. One or two reductase enzymes will be there in anabolic pathway. Now that reductase enzyme has to do reduction. Let's say A gets converted to A reduced. And now for this reduction, hydrogen is needed for reduction. Who will give this hydrogen? NADPH will give this hydrogen, right? So I say NADPH is used in reductive biosynthesis. That is for reductase enzyme, it is used in anabolic pathways. So can I say NADPH is produced from HMP? So HMP will convert NADP to NADPH. Now this NADPH is used in reductive biosynthesis pathways, making again NADP, and this NADP will be again used by HMP forming NADPH. So like this NADPH cycle will be occurring in cells. So now tell me what is the EC number of enzyme dehydrogenase. Dehydrogenase is doing oxidation. So here EC number will be 1. What is the EC number of reductase? Reductase EC number is also 1. It is doing reduction. I told you any enzyme which is doing oxidation reduction, that EC number is 1. Now tell me what kinase enzyme is doing. Kinase enzyme will do phosphorylation, you know that. To be very precise, I will say it will transfer phosphate. It will transfer phosphate. So EC number is 2, that is belonging to transferase category. There is one more enzyme, phosphorylase. Phosphorylase will also do the same thing, that it also transfers phosphate. Here also EC number is 2. But now tell me what is the difference between kinase and phosphorylase. Actually, kinase will transfer organic phosphate. Organic phosphate means the phosphate is present in some compound like ADP, ATP. So this is organic phosphate which is used by kinase. But phosphorylase will transfer inorganic phosphate. Inorganic phosphate like we write na? PI, inorganic phosphate. This is the free phosphate which is always present in the cells, right? So free phosphate which is not bound, that is inorganic phosphate, that is transferred by phosphorylase enzyme, and the organic phosphate that is in the form of ADP, ATP especially, that is transferred by kinase enzyme. Now there's one more enzyme which you should also see here on the same page because it is in comparison now. See the name phosphatase. You should be very clear with each and every basic term here. The enzyme is phosphatase. See, phosphorylase, I told you, it will transfer inorganic phosphate. But now I'm writing phosphatase. This phosphatase actually removes phosphate. And when phosphatase removes phosphate, then in this reaction, it is written like this. Water is used to break the phosphate, and this phosphate is released in the form of inorganic phosphate. So the EC number of phosphatase will be 3. That is, water is used to break the bond. So EC number is 3. So be careful. Phosphorylase EC number is 2. Phosphatase EC number is 3. Now see here, these 3 categories of enzymes, kinase, phosphorylase and carboxylases, they all use magnesium as cofactor. But you have to learn one exception, that is pyruvate kinase uses potassium more than magnesium. So if they ask you a question for pyruvate kinase, then you will mark potassium. If potassium not given, then mark magnesium, right? Now whenever kinases work, then either ATP is used or ATP is produced. ATP used means ATP is converted to ADP. ATP produced means ADP is converted to ATP, and this is very important thing, production of ATP in the body, and see how this ATP is getting formed. The enzyme is a kinase. Some substrate gets converted to product. Only one step occurs here at the level of substrate. Then and there only, immediately ATPs are formed at the level of substrate. This is known as substrate level phosphorylation, short form SLP, substrate level phosphorylation. There is no need of complex ETC for the formation of this ATP, that is substrate level phosphorylation. Very easy step which can occur here, but very few ATPs we are getting via substrate level phosphorylation. Most of the ATP we are getting via ETC, right? Now there's one trick for question solving. There are many questions asked on substrate level phosphorylation, and mostly they ask you the enzyme. Which of the following is enzyme of substrate level phosphorylation? Can I say enzyme of substrate level phosphorylation will always be some kinase, right? It will always be some kinase. So like a question is there, which of the following is not enzyme of substrate level phosphorylation? 3 options are some kinases. 4th option, which is not a kinase, that will be the answer. So this way you can easily solve. And the other method of ATP formation in the body is ETC. ETC is known as oxidative phosphorylation. This way, most of the ATPs are formed in the body, in the cells, but it is a very complex way of ATP formation, right? So substrate level phosphorylation is easy, but few ATPs are obtained by this. ETC is complex, but most of the ATPs in the cells are derived from ETC. See, when synthesis occurs in some reaction, 2 enzymes can be used. One is synthase, other is synthetase. Synthase, the smaller name, synthase. When synthase is used, then no ATP is required. But when synthetase, see the name? It is bigger name, synthetase. When synthetase used, then ATP is used to join the 2 compounds for the synthesis. So you can easily guess the EC number here. EC number in case of synthase is 4, that is lyase. But EC number in case of synthetase is 6, that is ligase category. ATP is used to make the bond. And in lyase category, EC number 4, ATP is not used to make the bond. So I can say the general line that all synthases are lyases, but we have few exceptions which we have to learn. One is nitric oxide synthase, which belongs to EC number 1, not 4. Glycogen synthase and citrate synthase, they belong to transferase category, EC number 2. And ATP synthase belongs to EC number 3, that is hydrolase category. So these 4 synthases are not lyases. This you have to learn. Now there are some reactions in body where carbon dioxide is added. When carbon dioxide is added, the enzyme is called carboxylase. Any carboxylase will use magnesium, I already told you, and there is a mnemonic for all the carboxylases. All carboxylases use ABC. This is a mnemonic where A stands for ATP, B stands for biotin, that is vitamin B7, and C stands for carbon dioxide. So all carboxylases use ATP, biotin, carbon dioxide and magnesium. So you will tell me the EC number here. What is the EC number of carboxylase? See, ATP is used and carbon dioxide is added and bond is formed. Carbon is added in some compound. So EC number will be 6 here, that is ligase category, because ATP is used to make the bond. Then there are few reactions in the body where carbon dioxide is removed. So that process will be called decarboxylation. This decarboxylation is further of 2 types, oxidative and simple decarboxylation. In oxidative decarboxylation, vitamin B1 is required. In simple decarboxylation, vitamin B6 is required. So I am summarizing now that B7 vitamin is used in carboxylation reactions, B1 vitamin is used in oxidative decarboxylation, and vitamin B6 is used in simple decarboxylation reactions. Now see the EC number. In case of decarboxylations. Oxidative decarboxylation EC number is 1 and simple decarboxylation EC number is 4. I will explain this. Why EC number is 4 here in simple decarboxylation because that carbon bond is broken without using water. That's why EC number is 4. And why EC number is 1 here in case of oxidative decarboxylation? Because whenever oxidative decarboxylation step occurs, then the enzyme is dehydrogenase. Enzyme is dehydrogenase and carbon dioxide is getting removed. When enzyme is dehydrogenase means oxidation occurring and removal of carbon dioxide. So this full is known as oxidative decarboxylation. There are many reactions of oxidative decarboxylation in the body. Especially it is there in link reaction and TCA cycle. Now let's see examples of simple decarboxylation. Simple decarboxylation, these reactions requires vitamin B6. Example is histidine to histamine, glutamate to GABA, tryptophan to tryptamine. And note here that all these are amino acids. Histidine, glutamate, tryptophan, all these are amino acids. And now from amino acids, we are removing carbon dioxide means we are removing acid. So from amino acid, when acid is removed, then we are left with amine. That's why the name histamine, tryptamine, amine is left here, right? And also tell me, from amino acid, if amino group is removed, if we are removing amino group, then we are left with acid, and this acid is having a keto group in this known as keto acid. So I summarize, from amino acid, if acid removed, then we are left with amine. If from amino acid amino removed, then we are left with keto acid, right? Now I will tell you one mnemonic kind of thing for coenzyme and vitamins. If in some reaction NAD is used, that means vitamin B3 is used. They are related. B3 is niacin and N is containing niacin here. Like other is, if in some reaction FAD used means vitamin B2 is used. They are related. It is flavin adenine dinucleotide and B2 is riboflavin, right? And if in some reaction coenzyme A is used means vitamin B5 is used. You can use it as a mnemonic for solving so many things. Now one more basic point here, that if a compound is having carboxy group or acid group, that compound will be having negative charge because carboxy ionized to give negative charge, and if a compound has amino group, then that compound will be having positive charge because amino ionized to give positive charge. So with this, I have completed the many basics required for understanding metabolism. In the next video, we'll be taking up glycolysis.